Okay. Good afternoon, everyone, and everyone online. There's Annie. Today is my great pleasure to share with you the regulatory considerations on DoseScale analysis in assessing pharmacodynamic equivalence. All right. Standard disclaimer, basically it says if you don't like what I said, blame me, not my agency. All right. Moving on. In the coming about 20 minutes, first we will have a quick review of the PD-BE study, as this is the foundation of this talk, and then focus on the main topic of today, dose-scale analysis for PD studies. And specifically, we'll talk about what is dose-scale analysis and when to use it, and how it's recommended in the product-specific guidance. And then we will discuss some considerations and challenges we encountered when we reviewed ANDA regarding dose-scale analysis associated with different aspects, including model fitting methods, Bootstrap implementation, missing data, and study. While I'll talk about these points, I will use case examples as illustration. I know most of you are familiar with this concept, but I couldn't help review it because it's the foundation of our talk. So in order to establish the therapeutic equivalence of generic drugs to its RLD, the generic drug needs to be pharmaceutical equivalent as well as bioequivalent. And drugs are considered bioequivalent if there is no significant difference in rate and extent of absorption at the site of action. To establish bioequivalence, four types of studies could be used, including comparative in vitro, PK, PD, and clinical endpoint studies. And today's talk's focus is on comparative PD studies. Right. So where will we use PD study to establish BE? I think the point was covered this morning in a couple of presentations about oral inhalation and nasal products. For the drugs that are systematically acting, PK approach can be used to measure the systemic exposure, then establish BE. But for locally acting products, where PK is the downstream of the therapeutic effect at the site of action, PK approach may not be sufficient. And for these cases, PD studies is more appropriate. And in the guidance, product-specific guidance, you could find PD studies recommended for oral inhalation drug products in combination with PK study and in vitro study as a weight of evidence approach to establish BE. And in locally GI tract acting products, and also topical corticosteroid and heparin injectables. So how are PD endpoint-based BE different from the PK endpoint-based BE? First, for PK endpoint-based BE, normally the exposure is proportional to the dose, and there's no exposure for placebo baseline correction. So the PK differences can be used to assess the difference between two products dose delivered by two products. And 90% confidence interval around the exposure could be used to establish BE. Differently, for PD endpoints, normally you don't see a linear relationship. Instead you see a nonlinear dose-response relationship where the dose does not increase proportionally with the dose. And there is often placebo effect that can be substantial. And 90% confidence interval around the PD response ratio, normally they're not used for establishing BE. This slide, I will show two examples where this nonlinear PD response relationship are observed. On the left is orlistat capsule. It is an intestinal lipase inhibitor. It's locally GI acting, so the systemic absorption is negligible. And the PK approach is not most appropriate to establish BE for this product. And clinical endpoint study could be an option, but its endpoint, which is the weight loss, can take time, so the study would be lengthy. Instead, PD study is more appropriate. And the PD endpoint for this product is the fecal fat excretion. And we observe a nonlinear relationship, and we see a plateau at the higher dose. On the right is a product we've discussed a lot in the morning. It's albuterol, a mere-dose inhaler, and it's locally acting. It's delivered to the lung, where the bronchodilator effect occurs. And there's limited relevance of systemic exposure to the amount of drug delivered at the site of action, so PK approach is not applicable. For this product, the PD study, there are two options. One is bronchoprovocation study, or bronchoconstriction study, and the other is bronchodilation study. Okay. So because of this non-linear PD response relationship, if we assess the differences in PD response scale, it may not directly reflect the differences in the relative bioavailability of the T and R products. To address this issue, DoseScale analysis is introduced to allow the assessment of the relative bioavailability on dose scale rather than the original PD scale. Because this analysis translates the nonlinear PD response to the linear dose, nonlinear PD scale measurement to the linear dose measurement scale. The result of it suggests equivalence, can be used to suggest the equivalence of the amount of drug reaching the site of action. So there are several components of DoseScale analysis. First, we have response from the placebo as a baseline, and then PD response from the reference product at the lower dose, and PD response from the reference product at the higher dose. From these, we could fit a dose response curve for the reference product. And then we have a test product response at the lower dose. So in this case, it's at 90 micrograms. And using the dose response curve for reference, so for the reference product we just fit, we could find the dose of reference product that would result in a response equal to that from the test product. It's an estimated value. And in this case, it's around 100 micrograms. Okay. And then relative bioavailability F could be calculated from the estimated dose, which is 100, by the actual dose, 90. And in this case, F is 1.11. And as the procedure suggests, this F could be considered as the ratio of the dose from the T and R that produces the same PD response. As you can see from that procedure, the essence of DoseScale analysis is Emax model fitting. So for Emax model fitting, there are two methods currently recommended at FDA in the guidance. And one, for the first method is used for the cases where you have one dose of test. Okay, equations are, you don't have to pay any attention to these equations. And these are just there in case you want to come back, refer to look for more details. And the second method is used in the cases where two dose levels of tests are available. And in this case, you simultaneously fit the curve, the dose response curve, for both reference and test. And for more details, again, you could follow this link to look at all the equations and all the more detailed description of this method. So to establish BE, 90% confidence interval of F should be calculated using a bootstrap procedure. First, a sample dose response sample set is generated using the bootstrap sampling with replacement. And then for each sample dose response data, the F is estimated by fitting Emax model using either method 1 or method 2. And then after that, FDA has recommended using Afron's biased, corrected, and accelerated BCA method to establish the 90% confidence interval of F. Again, follow the slide, follow the link for more details. And now we're back to the example, just 2 example drugs that I showed. So DoseScale analysis for PD study are recommended in several products, and these are the 2 example products. And this, there are a lot of details on this. This is basically to say what is the study design and what is the PD endpoint. And one thing to note is that for the orlistat, the BE criteria bound is from 80% to 125%. But for albuterol and levobuterol, they have a wider BE criteria bound from 67 to 150 because of the statistical and clinical considerations. All right. Okay. Moving on. For the Emax model fitting, there are several methods available, including the naive average and naive pool and nonlinear mixed effect modeling. And for the naive average method, the data from each individual are averaged so that there is only 1 data point per dose for each formulation. As the plot show here, there are only 3 dots, or actually 4 dots here. And for naive whole data method, we don't do any average. We keep the original data, but all the data from all the individuals are pulled together as if they come from the same individual. And then there's nonlinear mixed effect modeling method, where all the individual data are used to fit the model while incorporating the between subject variability. So for the NAD approach, naive average, you can see that by doing the average, you are actually actively reducing the available data. And there is no direct estimate of variability. And by the way, BSV stands for between subject variability. And if BSV is large, the estimation from this method could be biased. And if the individual has different amount of data or aberrant observation, the potentially biased results may be generated. And for MPD method, it is preferred compared to the NAD approach. But again, because you're treating all the data come from the same subject, if the between subject variability is large, there can be biased results. And their potential bias as the data come from non-standard designs can be pulled together. And for the nonlinear mixed effect modeling method, because it incorporates the between subject variability and residual unexplained variability. So it handles rich and sparse data and missing data well compared to the other 2 approach. And in the ANDA submission and also in our review, this method have been routinely used for Emax model fitting. In terms of bootstrap sampling, in the ANDA submissions, we've seen different ways of implemented. Just because it's a crossover study where for one subject, there can be multiple observation from multiple treatment arms. Just to give an example, if we have original data and where each subject were dosed placebo, reference at lower dose, reference at higher dose, and test at the lower dose, we've seen in some cases they resample the observation by ignoring the subject. And there are cases where they use the subject, they resample the subjects, but they only applied it for the reference data. Then there's this way of implemented to resample this subject. So for each subject that becomes the sample all the data, original data from all the treatment arms of that project, of that subject is remained. So this, the last one, we recommended to this last implementation method as it used the subject as the bootstrap sampling, where we remain all the data from the TNR for that specific subject. By doing that, we will maintain the correlation of the observation within the subject. So here, before I move on to the other two aspects, I want to do a quick summary of some practical considerations. So for fitting Emacs model, among the several available approaches, NLME approach is preferred as it incorporates between subject variability and is less sensitive to aberrant observations. And it has been routinely used in end of submission and review. And in the end of submission, we've seen the DoseScale analysis implemented in various kinds of software, including NOMEM, SAS, R, PFARM. And an internal investigation indicate that the results are generally consistent with the same model structure and parameter settings. And FDA has no requirements of which specific software you should use. Regarding computing the 90% confidence intervals of F, the recommendation is to resample original dose scale observations at subject level and to use minimize of 1,000 bootstraps. And we recommend following the bootstrap procedure in the guidance. And pre-specify modeling software and computation method for 90% confidence interval, in case you want to propose different ways of implement it. Okay. Moving on to another challenge encountered in the end of review is missing data. In this specific case, it's for the albuterol in the broncho provocation PD study. And the endpoint for this study is the PC20. And when it comes to missing data, now there is no consensus on how much portion of missing data is acceptable and would not cause bias in the B assessment. So some say that 10% is okay, but some say even 5% is too much. However, it is more important for the missing mechanism and the data pattern compared to the portion of the missing data that can affect the results. So there are several missing mechanisms: missing completely at random, missing at random, or missing not at random. Because there are always potential confounding factors that cannot be fully captured, so it's almost impossible to justify whether the missing data are truly missing completely at random. And crossover study design can be sensitive to missing data. For example, if you miss one period for a certain subject, that subject may lose self-reference. And subsequently, missing data may impact the F estimation in DoseScale analysis. Here, this is a case example of internal research project to investigate the impact of missing data on the F estimation. And here we simulate a 5-way 5-treatment crossover study with some assumption. And we purposely delete the data to create scenarios, different missing data scenario. We delete at random to create a missing at random case, and we delete only the lower end of the data distribution to create a missing not at random case. And then analyze the data using naive average approach and nonlinear mixed modeling approaches to see which one can recover the assumed true F. As the result suggested, in this case, if the data is missing at random, both methods seems to work equally fine. But when it's missing not at random, for example, for the first case where the true F is set as 0.96, and the B range past the the confidence intervals past the BE, the naive average method failed to capture that. So our tips learning from this case example is that when there are missing values not at random, nonlinear mixed effect model is less sensitive to missing value. But it should be pointed out, both of the methods have some deviation when it comes to the point estimate compared to the true F. So another challenge is about data imputation. So in this case, rather than a subject drop off the study and miss a value, miss data, in this case, the subjects, the missing data are generated because the subjects are non-responsive and they are not achieving 20% drop in FEV1 when they receive the maximum concentration of methacholine. So in this particular example, in this case, PC20 is not measurable, so it's missing. And the firm choose to impute a measurable value using the maximum concentration of methacholine. And unfortunately, they did a lot of imputation, the same value, maximum value, for both T and R treatment arms. So as you can see from this graph, the upper shows the without the imputation, and the lower shows with imputation. You can see after the imputation, the T and R differences significantly decreased. It's almost like by imputing the data, they're trying to make T and R more equivalent. And this is a concern because it might potentially bias the B equivalent, the B evaluation. So for this case, our recommendation is that if you're going to apply some data imputation method, you may want to specify that strategy in your plan, supported by adequate justification. And it's recommended that you evaluate whether the chosen imputation method is valid and sensitive in detecting the product differences. Okay, last case. This is kind of a unique case because in this pre-ANDA meeting request, the firm asked to conduct a bronchodilatation study. Well, in the majority of cases, a bronchoprovocation study is conducted. So in this case, because of the time, just I'll highlight. So our findings from this case example is that for bronchodilation study, depending on the study protocol and data, the DoseScale analysis for this kind of type of studies may be insensitive to detect the difference in relative bioavailability. Right. To summarize, today we talk about the DoseScale analysis and how to apply it. And so for DoseScale analysis, PSG reflects the agency's current thinking and recommendations. And towards a reliable DoseScale analysis, there are several aspects you could pay attention to. One is from the study design. You could submit a protocol for FDA evaluation before you initiate a pivotal BE study. And you could design a pilot study to refine your study design in terms of inclusion and exclusion criteria, power of a study. And for the data, if there's missing data, or potentially missing data, in the protocol you state how missing data will be handled. And for the modeling, if you're using alternative approach, you should provide sufficient justification for that. Okay, last, I think this becomes a recurring message that if you're proposing alternative method or some method that is significant deviate from the PSG, you should discuss with the agency. With that, I want to thank my colleague who worked on the projects I've spoken of in Office of Bioequivalence and Office of Research and Standard under OGD. And I thank you all very much for your attention.